Hi, and thanks for being here. This is the second segment on the physiology of the reproductive systems, and this is our last recorded lecture of the semester being spring 2020. And as this is the last lecture, it makes me long to be in the classroom with everyone. We have fun, especially when we talk about the reproductive system. And now I'm speaking into a microphone, and I miss you. I miss your faces, I miss your energy, and I wish it didn't have to end this way. And thank you for taking the journey with me. It's always more fun with you. And even though we're apart, it's still better with you than without you. So this is our last lecture, and we will cover slides 59 through 112, or the end of the reproductive systems. And so in this second segment, we will cover points 4 through 8. So our focus here will be on the female reproductive system. In segment 2, we'll focus on functions of the female reproductive system, and those functions include gametogenesis, which is the formation of gametes. And in the male reproductive system, we talked about spermatogenesis, which is the formation of sperm. In the female reproductive system, we will talk about oogenesis. Sometimes it's pronounced oogenesis. Oogenesis is the formation of eggs. We will also talk about the female reproductive cycle, and this is the monthly changes, a monthly sequence of events, that's a cycle, that occur in both the ovaries, so there's an ovarian cycle, and then there is also a uterine cycle. And the reproductive cycle is dictated, it's controlled by hormones produced by the hypothalamus and the anterior pituitary. So we'll talk about hormonal control of the ovarian cycle. We'll also talk a little bit about pregnancy, and we'll talk especially about how to avoid pregnancy, and we'll talk a little bit about lactation. So first we'll start with an overview of the anatomy of the female reproductive system. So unlike the male reproductive system, the female reproductive system is completely separate from the urinary system. And so let me show you the anatomy of the urinary system here. So let's choose green. This is the urinary bladder, and then this is the urethra. And it shares no common plumbing with the female reproductive system. In the female reproductive system, this is the uterus, and this is the vagina, a completely separate tube. And then, of course, this is part of the digestive system, the rectum and the anus. Here's a different view of much of the female reproductive system, where our gonads here are ovaries. Ovaries are paired. There's a right and a left ovary. And ovaries produce secondary oocytes, which go on to become eggs if they're fertilized. We'll talk a lot more about that in just a little bit. So the ovaries are the site of gamete formation, and they also produce an array of hormones, and the hormones that are most important for us are estrogens and progesterone, and we'll talk more about those when we get to the reproductive cycle. The uterine tubes here are not physically connected to the ovary, but they have these projections here known as fimbriae, and these fimbriae can beat rhythmically to generate local currents that draw the secondary oocyte that gets ovulated, passes through this ovarian tube or fallopian tube. As it makes its way, it migrates toward the uterus. The uterus is a muscular sac. We can view the layers of the uterus, the layers of the uterine wall. The endometrium is the deepest layer of the uterine wall. It's the interior lining of the uterus. And this is the structure that is built up and shed during the uterine cycle, which is part of the female reproductive cycle. And then this thick region of the uterine wall is called the myometrium. Myo means muscle. So the uterus is a muscular sac. This is smooth muscle here. And then the inferior portion of the uterus is called the cervix, and the cervix is where the uterus meets the vagina. And the vagina is a tube. It's a passageway for a penis, for example. It's also a passageway for semen. It's a passageway for menstrual blood. It's also sometimes called the birth canal because it's a passageway for a newborn baby. And the vagina, the walls of the vagina have rugae, which are these wrinkles in the mucosa of the vagina. And maybe you recognize that word rugae because we also have these wrinkles in the mucosa of the stomach. The lining of the stomach has rugae and the lining of the vagina has rugae. And in both cases, in both the stomach and the vagina, these rugae, they flatten out. As the stomach becomes filled with food, those wrinkles flatten out. And as the vagina becomes occupied with something like a penis or a baby or perhaps something else, those rugae flatten out. And this reminds me of many years ago in class, as I was talking about rugae, I saw somebody's face light up. And he shared with the class that his childhood dog was a Shar-Pei. It's one of those wrinkly dogs. And his childhood dog's name was Rugae. And he never understood the significance of that word, but he explained that his grandfather named the dog Rugae, and his grandfather was a physician. And so it was nice to see some pieces coming together for this young person. All right, so that's the basic anatomy of much, certainly not all, of the female reproductive system. So I hope you recall that gametogenesis is the formation of gametes, and gametes are egg and sperm, and spermatogenesis is the formation of sperm, and we covered that in the male reproductive system in the first segment. And today we're going to cover oogenesis, which is the formation of eggs. And that process begins in the ovary. The ovary is the female gonad. And in many ways, spermatogenesis is much simpler. It's almost just straightforward meiosis. And oogenesis is a bit more complicated than spermatogenesis. And these are 4 ways in which oogenesis and spermatogenesis differ. And I'm going to use these 4 points of departure as a framework for teaching oogenesis. So oogenesis and spermatogenesis differ in the timing of the onset. When do they begin in a human being? And they also differ in the number of gametes that result from meiosis. They also result in gametes that differ in quite an extreme fashion, that differ in size. And then when meiosis is completed, also differs significantly in the 2 different reproductive systems. So, when does it begin? In the male reproductive system, spermatogenesis begins around the onset of puberty. So somewhere 8, 9, 10, 11 years old, for the very first time in that young male's body, sperm begin to be produced. But in the female body, oogenesis begins while she is still a fetus. In the 3rd trimester of pregnancy, that female fetus is beginning oogenesis. And we'll take a look at what some of these words mean very soon. So when a baby girl is born, she has roughly 200,000 to 2 million primary oocytes. So that process of oogenesis has started. Those cells are in a phase of meiosis and put on pause. Meiosis is arrested. So following her birth, nothing more is happening to those oocytes until the onset of puberty. And then oogenesis resumes. So it begins during fetal development and it resumes at the beginning of puberty. And so here's a layout, a diagram of the process of oogenesis, and we'll just walk through it. So up at the top we have, you can think of oogonia as sperm stem cells. They are diploid, meaning they have the full genetic complement. They have 23 homologous pairs of chromosomes, meaning there are 46 individual chromosomes inside one oogonium. And then during fetal development, meiosis 1, the first meiotic division, begins, but is definitely not completed. So during fetal development, oogonia gives rise to primary oocytes. What process of cell division happens here? In going from oogonia to primary oocytes, it's mitosis happening here because the oogonium is diploid and the primary oocyte is diploid. And so this process must be mitosis. And then in going from the primary oocyte to the secondary oocyte, notice we have a change in the number of chromosomes. So this is meiosis 1. And meiosis 1 begins but is not completed during fetal development. So a female child is born with primary oocytes that have started meiosis 1, but then it's like the pause button is hit. And it turns out, and we'll look at these later, it turns out that these oocytes in the ovary, they're surrounded by a group of cells known as follicular cells. So this oocyte is situated inside a follicle, and these follicular cells make up the walls of the follicle. And these follicular cells send signals to the primary oocyte that allow that oocyte to remain paused in meiosis 1. So following birth, a female baby has between 200,000 and 2 million primary oocytes that have started meiosis 1, but they are in the pause state. It's been arrested. And so we fast forward until the onset of puberty, so 9, 10, 11, 12, 13 years old. She gets her first period. And so the ovarian cycle has started, and we'll talk a lot more about the ovarian cycle, but part of the ovarian cycle includes the continuation of oogenesis. So during the ovarian cycle, for a woman who is now in the reproductive phase of her life, a handful of these primary oocytes each month resume meiosis. And so meiosis 1 gives rise to a secondary oocyte as well as the first polar body. So in oogenesis, cytokinesis, which refers to the division of the cytosol between two cells during cell division, cytokinesis is uneven. So during meiosis 1, as well as meiosis 2, in oogenesis, each cell division, meiosis 1 and meiosis 2, produces one large cell and one small cell. And that very small cell is known as a polar body. As far as we know, it has no function other than to cart away and get rid of half of the chromosomes. So we think that in most cases, the polar bodies degenerate and they don't serve any purpose other than helping to get rid of that extra set of chromosomes. Remember, because our goal here is to create a gamete. And a fully formed gamete is haploid. It has just one set of chromosomes. So here we're looking at our first meiotic division, where we move from the primary oocyte to the secondary oocyte. And this happens during the ovarian cycle for a woman that's in the reproductive phase of her life. And so this secondary oocyte, it's big. It got most of the cytosol. It got the organelles like mitochondria and endoplasmic reticulum, etc. And so it is this secondary oocyte that begins the process of meiosis 2, the second meiotic division, but that gets arrested. So what gets ovulated each month is a secondary oocyte that has started meiosis 2, but then meiosis 2 was paused. And so that secondary oocyte gets ovulated. It gets ejected from the ovary. It gets swept up into the uterine tube. It's technically not an egg yet. Meiosis is not completed. It's a secondary oocyte. And so here we see ovulation occurring, and we have our secondary oocyte. Meiosis II is only completed if sperm penetration occurs. So if and only if this secondary oocyte gets penetrated by a sperm cell. So the acrosome that's present on the head of the sperm helps the sperm to burrow into that egg. Only then is meiosis II resumed and then completed. So here we're looking at sperm penetration followed by fertilization. The sperm penetration, the sperm digging into the egg, is the cue that allows meiosis II to be completed. And once again, we have unequal division of the cytoplasm. So during this cell division here, we form what's known as the second polar body. It's tiny, it gets just a little bit of cytosol, it's got that other set of chromosomes that we need to get rid of. We think that that second polar body degrades. We're not aware of any functional significance other than to carry away the extra chromosomes and get rid of them. And what we're left with is an ovum, that's an egg. Meiosis II is completed, this ovum has a haploid set of chromosomes, and then fertilization can occur. So this diagram is a little off. It doesn't distinguish between sperm penetration, which is when the sperm burrows into the egg, and then fertilization, a separate process, is when the sperm nucleus fuses with the egg nucleus. And so here we have what's really being shown here is sperm penetration. And then following fertilization, we end up with a diploid zygote. So the chromosomes that are brought by the sperm meet up with the chromosomes that were present in the egg, and now we have restored the full genetic complement, and we have a diploid zygote. So we're still talking about oogenesis, and we're still using that framework for understanding the ways that oogenesis differs from spermatogenesis as a way to further our understanding of oogenesis. So if you're a genetic female, if you're a biological female, at birth, you were born with between 200,000 and 2 million primary oocytes. They had already started meiosis I, but the molecular pause button was hit. And as you grew, as you went through infancy and childhood and hit puberty, many of those primary oocytes degraded, and that is known as atresia. And so the number of oocytes that degrade or undergo atresia varies pretty widely among individuals, but on average, when a young woman hits puberty, she has roughly 40,000 primary oocytes that are arrested in meiosis I. And every month, a handful of those will resume meiosis I, and typically just one will get ovulated as a secondary oocyte. And so if you sum up her total reproductive years, so every ovarian cycle that she has from puberty to menopause, on average, it's roughly 400 of those primary oocytes give rise to secondary oocytes that get ovulated. And then menopause hits, and it is a myth that menopause occurs because the ovaries run out of eggs. That's not true. There are still functional primary oocytes inside the ovaries of menopausal women. Rather, menopause occurs due to changes in hormone production, hormone secretion by the hypothalamus and the pituitary glands, and we'll talk more about that in a little bit. So another contrast between spermatogenesis and oogenesis is the number of gametes that result from meiosis. So if we start with a primary spermatocyte, and we'll contrast that to starting with a primary oocyte. If we start with one primary spermatocyte, it will undergo meiosis to produce four individual sperm cells. And that's the basics of meiosis, right? One parent cell divides twice to produce 4 daughter cells. In this case, the daughter cells are sperm. In oogenesis, one primary oocyte will undergo meiosis, and the outcome is one ovum. Only one egg results because we have that unequal division of the cytoplasm. We have the generation of 2 polar bodies and the outcome of just one egg or one ovum. So here we can do the side-by-side comparison of, on the left is spermatogenesis and on the right is oogenesis. Let me walk you through spermatogenesis first. So spermatogenesis is almost just basic meiosis. We have our first meiotic division where we have a primary spermatocyte that gives rise to 2 secondary spermatocytes. And then each of these secondary spermatocytes undergo the second meiotic division to give rise to 4 equal spermatids. And then those spermatids each differentiate, they undergo spermiation, they differentiate to become 4 sperm. So that's spermatogenesis. Oogenesis looks quite a bit different. We start with a primary oocyte that undergoes meiosis 1 to give rise to a secondary oocyte, but just one secondary oocyte, because over here we have our first polar body. This secondary oocyte gets ovulated, and only following sperm penetration does that second oocyte undergo meiosis 2 to form this one ovum. And this one ovum will now undergo fertilization, where the nucleus from the sperm fuses with the nucleus from the egg to form a zygote. The size of the gametes. So sperm are the smallest cells in the human body, and an ovum is the largest cell in the human body. It's reportedly visible with the naked eye. I looked for years and years and I was never able to see it. That's a joke. It is reportedly visible with the naked eye. And the reason for the size difference is during the production of sperm, a process known as spermiation occurs. That's just the differentiation of sperm. And during that differentiation process, much of the cytoplasm and the organelles are shed. They're discarded in order to create this tiny cell. It's very streamlined. It's basically DNA with a propeller and mitochondria to fuel that propeller. And then an egg cell is the largest cell in the body. It has lots of cytoplasm, lots of mitochondria and other organelles. And that cytoplasm and those organelles will become part of the resulting zygote should fertilization occur. And then the fourth difference between spermatogenesis and oogenesis is, of course, when that second meiotic division occurs. So in the male body, during spermatogenesis, meiosis 2 is completed inside the seminiferous tubule, and those resulting gametes, those fully formed sperm, migrate out of the seminiferous tubule and into the epididymis. They're full-on gametes ready to go. But in the female reproductive system, oogenesis is only completed following sperm penetration. So the head of the sperm burrows into the outer layers around the egg, and only then is meiosis 2 completed. Only then do we form what is, biologically speaking, technically a gamete. It's got the haploid complement of chromosomes. And if sperm penetration doesn't happen, then meiosis 2 is never completed, and she gets her period, and that secondary oocyte flows out of her body as part of menstrual discharge. So we've looked at this picture before. We can use it to reinforce the concepts that we've been talking about before we move on. So I'll remind you that oogenesis begins during fetal development, and a baby girl fetus, so she's still in the womb, has primary oocytes that have started meiosis 1, but then the metabolic pause button is hit, and she goes through early childhood with 200,000 to 2 million primary oocytes, and some of them start to resume development at the onset of puberty. And following puberty, she will experience the monthly reproductive cycle, which we'll talk about in just a little bit. So during the ovarian cycle, each month a handful of those primary spermatocytes resume meiosis, but only one typically completes the first meiotic division to become a secondary oocyte. And this secondary oocyte starts meiosis 2, but that meiosis 2 is arrested, the pause button is hit, and the secondary oocyte pops out of the ovary and moves through the uterine tube toward the uterus. So the secondary oocyte is what gets ovulated. If sperm penetration does not occur, the secondary oocyte never finishes meiosis 2, and it leaves the body with her next period. But if sperm penetration does occur, then meiosis 2 resumes and the second meiotic division occurs. And remember, these meiotic divisions are unequal in terms of the way the cytosol is distributed between the two daughter cells. So we have the formation of an ovum. This is technically an egg. It's a gamete. It has half the number of chromosomes. And then we have our second polar body over here. It's tiny. It's just a way of getting rid of those extra chromosomes. And then here we see the process of fertilization occurring as the nucleus from the sperm fuses with the nucleus of the egg to form a zygote. So the female reproductive cycle is a repeating sequence of events that occurs roughly every 28 days, and we'll use 28 days as the example cycle here, but it varies quite a bit among women. Some women have shorter cycles. They might have a reproductive cycle that's perhaps 19 days long, meaning every 19 days she gets her period, or she might have a longer cycle. Maybe every 35 days she gets her period. And then some women have irregular cycles, which makes their period more difficult to predict, and that's fairly common as well. So we're just going to talk about the woman, the female body that has a regular 28-day reproductive cycle. So the reproductive cycle involves two different cycles. There are monthly events that occur in the ovaries, and there are monthly events that occur in the uterus. And we'll study these separately, and of course they're interrelated, and we'll talk about how they are connected. And I hope you're thinking about cell signaling at this point, right? Because the ovaries and the uterus, they're two different organs. And how are these organs syncing up their cycles? And it is due to hormonal signals. In covering hormonal control of the ovaries, you'll recognize some familiar players because we talked about them and their role in the male reproductive system. We will encounter the hypothalamus and the pituitary gland, and of course the gonad. In the male reproductive system, it's the testis. In the female reproductive system, it's the ovary. So, under the influence of GnRH, not shown here, but I could draw it in, this would be the hypothalamus over here, and the hypothalamus secretes gonadotropin releasing hormone, GnRH, and we talked about that when we covered the male reproductive system. It's the same setup in the female reproductive system. The hypothalamus secretes gonadotropin releasing hormone, or GnRH, and that triggers the anterior pituitary to secrete FSH and LH. FSH is follicle stimulating hormone, and LH is luteinizing hormone. And both these hormones, we'll talk about them collectively, just like we did in the male reproductive system. I didn't differentiate, I did not distinguish these two hormones. I talked about them collectively as being responsible for triggering testosterone production and sperm production. I'll do the same thing in the female reproductive cycle. Please know that if you go on to study this in greater detail down the road in another course, that FSH and LH do have independent functions. Cells respond differently to each of these hormones, but I'll be talking about them collectively. So FSH and LH secreted by the anterior pituitary influence the ovarian cycle, this monthly sequence of events that occur in the ovaries. And the ovaries are the source of oogenesis, the site of oogenesis, and the ovaries are also an endocrine structure. And the ovaries produce several different hormones. We'll talk about the estrogens and progesterone, and estrogens and progesterone govern the uterine cycle, the monthly sequence of events that occur in the uterus. So in short, the hypothalamus controls the anterior pituitary, hormones from the anterior pituitary control the ovaries, and hormones from the ovaries control the uterus. And of course, this hormonal communication system operates through negative feedback systems. So we looked at an image very similar to this when we covered the male reproductive system. And what did we just talk about in the previous slide? We talked about, let me change colors here. Let's pick blue because there's not a lot of blue on this slide. So we just talked about, on the previous slide, we talked about the hypothalamus secreting gonadotropin releasing hormone, and that affects the anterior pituitary, and in response to GnRH, cells in the anterior pituitary increase their secretion of LH and FSH, and those hormones influence the behavior of the ovaries. In response to these hormones, and we'll talk more about what that looks like, the ovaries increase their production of estrogens and progesterone, and these hormones influence what happens in the uterus. And so that's what's happening on the left side of this diagram. The feedback mechanisms are happening on the right side of this diagram. So there are sensitive cells in the anterior pituitary and the hypothalamus. They're constantly monitoring circulating levels of estrogen and progesterone. And by circulating, I mean it's present in the blood. A circulating hormone travels in blood. And as we'll see very soon, circulating levels of estrogens and progesterone vary throughout the monthly cycle. They're higher at some points and lower at other points. And so there are estrogen and progesterone sensitive cells in the anterior pituitary as well as the hypothalamus. And they're constantly monitoring, well, how much of these hormones is present now? How about now? How about now? And when these hormones are abundant, it'll exert a negative feedback. It'll trigger a negative feedback intervention on the production of both gonadotropin releasing hormone and LH and FSH. So when estrogen and progesterone levels are high, the hypothalamus responds by decreasing its production of gonadotropin releasing hormone, and the anterior pituitary responds by decreasing its production of luteinizing hormone and follicle stimulating hormone. So I mentioned that the female reproductive cycle includes two cycles, the ovarian cycle and the uterine cycle. So we're going to examine them separately and then we'll put them together. So for this slide and the next few slides, we'll be thinking about what happens in the ovaries. We're going to ignore the uterus for a little while. So in the ovaries, we have the following events that occur as part of the ovarian cycle. The oocyte matures. So meiosis is going to resume to produce that secondary oocyte. And then that oocyte sits in a little pocket called a follicle. That oocyte is surrounded by several layers of cells known as follicular cells, and that whole structure is known as a follicle. Well, it turns out that those follicles also start to develop. Those cells undergo mitosis, they start producing hormones, and the hormones that those cells start to produce are the estrogens. And as that follicle develops and as that oocyte goes on to become a secondary oocyte, that secondary oocyte is ovulated. It is ejected, physically ejected across the wall of the ovary into the pelvic cavity, and it gets swept up into the uterine tube. So that's ovulation. And then following ovulation, that empty follicle, it goes on to develop into a brand new endocrine structure called the corpus luteum. And that corpus luteum, those cells begin to produce progesterone. And so these are the components of the ovarian cycle that we're going to look at in more detail. The primary focus of this image is follicular development. So follicular development occurs as part of the ovarian cycle. And I'll remind you that a follicle, each one of these images shows a follicle in a different stage of development. A follicle includes the oocyte plus the surrounding follicular cells. Sometimes they're called granulosa cells. And so two things are occurring here as we follow the arrows. Number one, this oocyte, that primary oocyte resumes meiosis and comes to form the secondary oocyte. So in this mature follicle down here, this cell here is the secondary oocyte, and that's what will be ovulated. The second series of events that are occurring here is follicular development. So surrounding the oocyte here are follicle cells. They're in gold, and they start to divide by mitosis, and the follicle gets larger. And these follicular cells start to produce estrogens. So the larger the follicle, the more estrogens are being produced. So her circulating estrogen levels are going up. And during follicular development, there's a stage in which this fluid-filled cavity known as the antrum forms, and that antrum grows larger and larger. Here's a fluid-filled cavity, and it surrounds the secondary oocyte. And the secondary oocyte will eventually protrude. It'll bulge out of the side of the ovary, and the wall of the ovary will rupture. And that will allow for the release of the secondary oocyte plus the cells that surround the secondary oocyte. So there are these granulosa cells that surround the secondary oocyte, and all of that gets ovulated. So here we're looking at the same concepts, but portrayed in a very different artistic representation. So over here, we see the primordial follicles that we saw on the previous image, and we see follicular development occurring throughout the days of the ovarian cycle, day 1, day 2, day 3, day 4. Before I go any further on this image, I want to be very clear that the follicles do not migrate around the ovary. They stay in place, they mature in place, and ovulation occurs right there. So this diagram could be a little misleading. The follicles mature in place, they don't migrate around. So two things are happening here. The follicle is developing and meiosis is resuming. So remember, in this primordial follicle, these primordial follicles here, they've been hanging out in that ovary ever since she was born, and now it's their turn. It's their turn to develop. It's their month. And a handful of these primordial follicles undergo follicular development, and they compete. Who can develop the fastest? And the fastest, the largest follicle actually sends out signaling molecules to inhibit the development of other competing follicles. So there's this competition, and typically every month just one follicle wins, and she ovulates this one secondary oocyte here. And I like this image here. This is a photomicrograph of imminent ovulation. So this is that secondary oocyte bulging off of the wall of the ovary. Following ovulation, this empty follicle goes on to become an endocrine structure known as the corpus luteus. The corpus luteus produces progesterone. So after ovulation, her circulating levels of progesterone increase as the corpus luteum develops and starts producing progesterone. The corpus luteum has an expiration date. It will only produce progesterone for approximately 2 weeks, and then it begins to degenerate and becomes something known as the corpus albicans, the white body. And the corpus albicans no longer produces progesterone. So the corpus luteum caused her circulating progesterone to increase, and as that corpus luteum degenerated, her circulating progesterone levels decrease. And as we'll see, that drop in progesterone is the reason for the beginning of her period. This is what a mature ovarian follicle looks like through a compound light microscope. So this whole thing is the follicle. This right here, this is the oocyte. And this is the nucleus of that secondary oocyte here. So it's arrested in meiosis II. And then these are granulosa cells, sometimes they're called follicular cells, and they will be ovulated along with the secondary oocyte. These follicular cells here are producing estrogen, so her estrogen levels are fairly high at this point in the cycle. This follicle will rupture, and this whole structure here, the oocyte with the surrounding granulosa cells, will be ovulated. This is taken using an endoscope, so a camera into her pelvic cavity. This is her ovary. And then this is a bulging follicle. So this is about to rupture. When it ruptures, that oocyte and the surrounding granulosa cells will be released into the pelvic cavity. We're going to spend a lot of time with this image. And let me give you a basic overview of what's being shown here. So up top, I hope you recognize the hypothalamus and the anterior pituitary. One controls the next. And then down below, we see the events that occur during the ovarian cycle. And the events of the ovarian cycle control the events that occur during the uterine cycle. I've blocked out the uterine cycle for now so that we can focus on the other items here, and we'll add the uterine cycle in later and pull it all together. And then finally down below, we have a timeline. And the cycle that we're using as an example cycle is 28 days long. And down here we see the names of the phases of the reproductive cycle: the menstrual phase, the pre-ovulatory phase, ovulation, and the post-ovulatory phase. And notice that we start with day 1 is the menstrual phase. And on this diagram, the end of the cycle is here, day 28. And on this diagram, we loop back around with days 1 and 2 again. So going up to the top of the image, I'll remind you that the hypothalamus controls hormone production by the anterior pituitary. And in response to gonadotropin releasing hormone, GnRH, the anterior pituitary secretes FSH and LH. And you can tell by the blue and orange arrows drawn here that FSH and LH play distinct roles in controlling the ovarian cycle. That's true. I will talk about FSH and LH together, even though in reality they do have distinct roles, but collectively they dictate the events that occur during the ovarian cycle. During the ovarian cycle, we have follicular development, and we also have meiosis, the resumption. Meiosis resumes. We have ovulation, we have the formation of the corpus luteum, and we have the degeneration of the corpus luteum. So let's walk through that. So at the end of the previous month, so days 26, 27, 28, follicular development in the ovaries begins anew. And we have a handful of primordial follicles that begin to develop. Meiosis resumes, so that primary oocyte completes meiosis 1 to become a secondary oocyte, and the surrounding follicular cells multiply by mitosis. They become more numerous, and they begin to secrete estrogens. And as follicular development continues, we get more and more follicular cells secreting more and more estrogens. And as we'll see, those estrogens play a role in controlling the uterine cycle. So we get one mature follicle that then ovulates. Here's that secondary oocyte surrounded by what's known as the corona radiata, the radiant crown, which makes me want to barf in terms of terminology. It's a terrible name. And so the secondary oocyte is ovulated, and this empty follicle here goes on to become the corpus luteum, or yellow body. And the corpus luteum begins to secrete progesterone as well as estrogens. And this corpus luteum will secrete these hormones for approximately 2 weeks. And then the corpus luteum degrades to become the corpus albicans. And to the best of our knowledge, the corpus albicans plays no role. It's just inert tissue. And so the hormones that are produced during the ovarian cycle, first estrogens, increasing levels of estrogens, followed by progesterone along with estrogens, followed by a significant drop in progesterone and estrogen. Those differing hormone levels control the uterine cycle, which is the next thing that we'll talk about. So the uterine cycle will focus specifically on what's going on in the uterus, which is a completely different organ from the ovaries. So remember, the uterus is a muscular sac, and the deepest, most layer of the uterus is called the endometrium. And that is a layer that increases in thickness during the uterine cycle, and then during menstruation, it's shed. That endometrium, most of that endometrium falls off and passes out of the body through the vagina during her period. The building up of the endometrium, the thickening of the endometrium, is in expectation of the arrival of a zygote. So fertilization typically occurs inside the uterine tube, and then that zygote begins to divide by mitosis and becomes an embryo. And that embryo, the very beginning of a pregnancy, that embryo migrates into the uterus and implants itself into the uterine wall, into specifically the endometrium. So the functional purpose of the endometrium is to be a hospitable place for the developing embryo. If that embryo doesn't arrive, then menstruation occurs. That endometrium degrades and it literally sloughs off, and she gets her period. The events of the uterine cycle, what happens with this endometrium as it gets thicker and then it sloughs off, and then it gets thicker and it sloughs off, that repeating monthly cycle of events, that's governed by hormones that are produced during the ovarian cycle, changes in circulating levels of estrogens as well as progesterone. So these are the phases of the uterine cycle. The menstrual phase is typically defined as day 1 to 5. She's menstruating. So the first day of her reproductive cycle, day 1, it's just a convention to call that the first day of your period. The next phase is called the pre-ovulatory phase. That's a good name. It's the phase that's before ovulation, and it's roughly days 6 to 13. The ovulatory phase is roughly day 14, and that's the day on which she ovulates. And then our last phase is called the post-ovulatory phase. Following ovulation, it stays roughly 15 to 28. So here's that same diagram, only now I've covered up the hypothalamus, the pituitary gland, and the ovarian cycle. And what I've left showing are the hormones that result from the ovarian cycle and the events that occur in the uterus over the 28-day monthly cycle. So let's look at this stratum functionalis and the stratum basalis. These are the layers of the endometrium. So remember, the endometrium is the deepest layer of the uterus. It is lining the lumen of the uterus. And the endometrium has these two layers, the stratum functionalis and the stratum basalis. The stratum basalis is permanent. It's represented sort of by that dark pink horizontal band. The stratum basalis doesn't change and does not change in thickness over the monthly cycle. It's the permanent layer that gives rise to the stratum functionalis. The stratum functionalis changes in thickness over the monthly cycle. And we can see, starting in the menstrual phase, we start down here, we see that the stratum functionalis is decreasing in thickness, and this is because it's being shed, it's being sloughed off. She has her period. And those cells, that mucus, there's blood vessels, there's blood, that's all being sloughed off and exiting our body through the vagina. And so her period ends and we move into the pre-ovulatory phase, and under the influence of estrogens coming from the ovaries, the endometrium rebuilds. Cells undergo mitosis, new blood vessels grow, new endometrial glands grow. So we have this thickening of the endometrium during the pre-ovulatory phase. Down here, the artist took care to include the growth of new blood vessels. That's called angiogenesis. And then in blue, I believe the artist is representing endometrial glands. And these glands develop anew each month, and these glands are responsible for secreting glycogen. And I hope you recall that glycogen is a polysaccharide. It's a storage form of glucose, and that glycogen that's secreted by these endometrial glands will be used as a fuel source for an embryo, should an embryo arrive and implant. One of the things that makes this endometrium a very hospitable environment for the flourishing, for the development of that embryo, is the fact that there's this abundant supply of glycogen, a fuel, being secreted by these endometrial glands, shown here in blue. All right, ovulation occurs here, and that means that the secondary oocyte and the surrounding granulosa cells are ejected into the pelvic cavity, and that secondary oocyte is swept up into the uterine tube, also known as the fallopian tube. And let's see, in this diagram, apparently fertilization and implantation do not occur. So she doesn't get pregnant this month. Woohoo! She doesn't get pregnant. And we're in the post-ovulatory phase where the endometrium is still lush. It's still well endowed with blood vessels and glands in expectation of this developing embryo. And what maintains the endometrium, what allows it, what prevents her from getting her period, is the production of progesterone and estrogens that are, of course, arising from the corpus luteum. So the corpus luteum is essentially saying to the uterus, hey, there's one on the way, don't get your period. I ovulated, you could be pregnant, don't get your period. But as you recall, that corpus luteum has an expiration date. It begins to degenerate, and after about 2 weeks, it stops putting out progesterone and estrogen. And due to that drop off in hormones, she gets her period. So we're back to day 1 with the first day of menstruation, and the cycle begins again. In the last image, I hope you recall that during the pre-ovulatory phase, the developing follicles in the ovaries are producing increasing amounts of estrogens. And those estrogens are what cause the endometrium to grow thicker, and that's part of the uterine cycle. So during the post-ovulatory phase, that ruptured follicle in the ovary becomes the corpus luteum, and that corpus luteum begins to secrete estrogens and progesterone. And it is these hormones that maintain the endometrium, that prevent her from getting her period. But the endometrium will expire, and that's going to lead to the onset of menstruation. Here we're looking at a uterus in the post-ovulatory phase of development. So the stratum functionalis is thick. It is filled with endometrial glands and blood vessels. Those endometrial glands produce glycogen as well as other nutrients in expectation of an embryo that will dig itself into this stratum functionalis and then be fed essentially by the glycogen and other nutrients that are secreted by these endometrial glands. If implantation does not occur, she doesn't get pregnant, woohoo, she doesn't get pregnant, then the drop off in circulating hormone levels, the drop off in estrogens and progesterone cause these blood vessels to become occluded. They stop passing blood through them, essentially cutting off the blood supply to the stratum functionalis. In the absence of a blood supply, this tissue starts to degenerate. The cells detach from one another, and the entire stratum functionalis is discharged through her vagina. So here's that same diagram we've been working with, only nothing is blocked off, and we can put all the components together. What's the role of the hypothalamus? What's the role of the anterior pituitary? What are the events that occur during the ovarian cycle? What hormones are produced during the ovarian cycle, and when, and by what? How do these hormones influence the uterine cycle? What are the phases of the uterine cycle? How do these hormones contribute to determining buildup of the endometrium versus shedding of the endometrium? So I hope you have a basic understanding of these types of questions by the time we finish the female reproductive system. If implantation does not occur, again, that corpus luteum will expire and it will stop producing progesterone as well as estrogens. And this declining state of circulating hormones is the trigger for the beginning of her period. We can use this diagram to examine changes in circulating hormone levels over the 28-day reproductive cycle. On the vertical axis is hormone concentration, and on the horizontal axis are the 28 days of this typical, quote, typical female reproductive cycle. And in different styles, colors of lines, we see hormone levels for 4 different hormones: FSH, estrogens, LH, and progesterone. Here are a couple of questions. Which hormones, of the 4 hormones shown here, which are secreted by the anterior pituitary? Which are secreted by the ovaries? I hope you correctly identified that FSH and LH are secreted by the anterior pituitary, and estrogens and progesterone are secreted by the ovaries. So again, the anterior pituitary controls the ovaries, and the ovaries control the uterus. What I'd like to focus on in this diagram are changes, monthly changes in estrogens and progesterone, along with this very obvious spike in luteinizing hormone. So let's take a look at what happens with estrogens and progesterone over the 28-day reproductive cycle. So let's start here. She has her period. She has her period because of the events that occurred during the previous month. The corpus luteum degenerated into the corpus albicans, and the corpus albicans doesn't secrete estrogens or progesterone. So looking at the menstrual phase of the reproductive cycle, let's see, estrogens are represented by the green line, and progesterone is represented by that maroon, that dark red line. So during the menstrual phase, circulating levels of these 2 hormones are low. And in fact, that's why she's menstruating, because the corpus luteum degenerated and these circulating hormone levels are low. So let's move into the pre-ovulatory phase here. During the pre-ovulatory phase in the ovaries, a handful of follicles are growing larger. those follicular cells surrounding the oocytes, they're dividing by mitosis. They're becoming more numerous and they're secreting estrogens. And as those follicles continue to increase in size, more and more estrogen is being produced. So during the pre-ovulatory phase, we have this rise in circulating levels of estrogen. And that estrogen is going to cause the endometrium to thicken. Here we have ovulation. Ovulation occurs roughly on day 14. And what triggers ovulation, what causes a weakening of the wall of the ovary, is this spike in LH that occurs here. So this spike in LH is known to allow or trigger, cause ovulation. And then see that LH levels decline pretty rapidly following that. So she's ovulated. Due to the abundant secretion of estrogens, we can assume that her endometrium is thick, and we're in the post-ovulatory phase here. So she's ovulated. So that one lone mature follicle that's now empty, it degrades. And so immediately following ovulation, her estrogen levels decline. But look at this, that corpus luteum very quickly starts to produce progesterone. So progesterone levels go up. And eventually her estrogen levels start to rebound a little bit because the corpus luteum also produces estrogens. So we're in the post-ovulatory phase. Progesterone as well as estrogens are responsible for maintaining that thick post-ovulatory endometrium in expectation of implantation. But it looks like we can tell from the hormone levels here, specifically the estrogen and the progesterone, we can tell that implantation did not occur. The corpus luteum degraded, it stopped putting out progesterone and estrogen, and this pretty swift decline in progesterone here is what triggers menses, the onset of menstruation. So far, I've been talking about the situation that happens most of the time. Implantation of an embryo does not occur. What happens if it does occur? What happens if that secondary oocyte is penetrated by a sperm, fertilization occurs, and that developing embryo makes its way into the uterus and buries itself in the stratum functionalis of the endometrium? What then? Why doesn't she get her period? That's an early sign of pregnancy. It's not always the first sign of pregnancy. It's not necessarily reliable, but it's a very common first sign of pregnancy. Her period is late. She doesn't get her period. Why? Well, that implanted embryo sends out its own set of signaling molecules as if to say, I'm here, don't get your period, I'm here, I made it. And so that signaling molecule is called human chorionic gonadotropin, or HCG. And it is a hormone that's produced by an embryo. So this tiny collection of cells that have implanted into the endometrium. And what HCG does, it's a signaling molecule, it's a hormone, and the target tissue of HCG is the ovary, specifically the cells in the corpus luteum. The cells in the corpus luteum will extend their life under the influence of HCG. So following implantation and the secretion of HCG by the embryo, the corpus luteum does not degenerate. Rather, it continues to produce progesterone and estrogens, which serve the purpose of maintaining that stratum functionalis, that endometrium. So under the influence of HCG, she does not get her period. Here we're looking at the process of ovulation, sperm penetration, fertilization, and the beginning of embryonic development. So Here we see ovulation. This is a secondary oocyte surrounded by granulosa cells. Here we see sperm penetration and fertilization. Can you see the little tiny sperm there? So the smallest cells in the human body meet the largest cell in the human body. So during fertilization, the nucleus of one sperm fuses with the nucleus of the ovum. And here we have a zygote. This is the resulting single cell. How many chromosomes are in this cell, in a zygote? 46. You're right, 46. There are 23 pairs of chromosomes in this zygote. Is it diploid or haploid? It's diploid. You're right. And so while this fertilized cell, while this zygote is traveling through the uterine tube, the first stages of embryonic development begin. It's called cleavage, where one cell divides to become two cells. Is this type of cell division, the first cleavage, is this type of cell division, is this mitosis or meiosis? It's mitosis. This is a diploid cell, the zygote is a diploid cell, and then all of these resulting cells during embryonic development, they're all diploid. Remember, mitosis is the vastly more common type of cell division in the human body, and meiosis is only for the production of gametes, egg and sperm, in the human body. And we're far from that in this pregnancy. So we have this very early embryo traveling through the uterine tube and into the uterus. This embryo will then connect with the stratum functionalis and release enzymes that degrade the stratum functionalis so that this embryo can anchor itself into the uterine wall, into the stratum functionalis. This process is called implantation. And during implantation, cells in this embryo are secreting human chorionic gonadotropin. And that's a signaling molecule whose target is the corpus luteum in the ovary. So signaling molecules, let me try to change color here. Signaling molecules arising from this embryo are influencing this corpus luteum. And in response, this corpus luteum will continue to produce progesterone and estrogens. It doesn't expire. And so this embryo right here is saying, hey, I'm here, I'm here, I'm here, don't get your period. And the reason she doesn't get her period is because this corpus luteum continues to put out estrogens and progesterone, maintaining this stratum functionalis. So you're in a position to understand how a home pregnancy test works. So a home pregnancy test, there are a variety of devices shown up top. What they all have in common is they test urine for the presence of human chorionic gonadotropin. So the only time HCG is present in an adult human body is when she's pregnant or has recently been pregnant. So how this works is there are reagent strips. One reagent strip serves as a control. The other reagent strip serves as a test. And the common outcome is when both of them turn pink, it means she's pregnant. If only one of them turns pink, it's the control. And it means that the test is working, but she's not pregnant. There's no human chorionic gonadotropin present. So how do we avoid getting pregnant? The most obvious way to avoid pregnancy is to not have sex in a way that allows semen into the vagina. All sorts of ways to have sex. If you don't want to get pregnant, one way to do that is to have sex in a way that doesn't allow semen anywhere near the vagina, or don't have sex at all. The variety of ways shown here fall into several categories. We'll talk about natural family planning. So what this means is, and sometimes it's referred to the rhythm method, is to avoid having vaginal penetration sex during times of the month when she is most likely to get pregnant. So it's around that ovulatory phase, several days before ovulation, several days after ovulation. If you avoid having vaginal penetration during that time of the month, it decreases the likelihood of a pregnancy. A second category here is barrier methods. So a barrier method prevents the sperm from contacting the egg. And so these condoms here, female and male condoms. The condoms act as a barrier that block the passage of sperm to the egg. And then the rest of the items here are all hormonal. Well, let me talk about IUDs first. An IUD is called an intrauterine device, and it's a little contraption that is implanted by a physician. It's implanted into the uterus, so it comes to occupy the lumen of the uterus. And it makes the uterine less hospitable to an embryo. So should fertilization occur, it makes implantation unlikely. And some IUDs are hormone-free, so it's just a mechanical device. And other IUDs also release hormones that improve its contraceptive ability. And then the rest of these methods shown here are all based in hormonal regulation of the female reproductive cycle. The hormonal methods of birth control use some combination of estrogens and progestin to influence the pituitary secretion of LH and FSH. So progestin is a progesterone agonist. Progestin is a signaling molecule that mimics progesterone. It binds to and activates progesterone receptors. And so for all of the hormonal methods of birth control, we use exogenous signaling molecules to influence the female reproductive system. And remember, exogenous means from outside the body. Hormonal methods of birth control are typically a combination of estrogens and progestin, or it could be progestin only. So these are exogenous hormones that take advantage of the negative feedback system for hormone production. So let's say the person is on a birth control pill, shown here. Every day for most of the month, she takes a pill, and that pill contains estrogens and progestin. And so that artificially elevates levels of these hormones in her blood. Remember, these hormone levels, we talked about this before, these hormone levels feed back on both the pituitary as well as the hypothalamus. And so in response to these elevated circulating levels of hormones, the hypothalamus secretes less gonadotropin releasing hormone. Oh, there's already plenty of estrogens and progesterone. It's the wrong time of the month. I shouldn't secrete GnRH. Simultaneously, the anterior pituitary also responds to these elevated levels of estrogens and progesterone. The anterior pituitary reduces its secretion of LH and FSH. With a lower level of LH and FSH being secreted, that interferes with the ovarian cycle. So follicular development doesn't occur. Ovulation doesn't occur. It's essentially tricking the endocrine system, tricking the reproductive system into thinking that it's in a different phase of the reproductive cycle than it's actually in. And it inhibits the production of pituitary hormones that then goes on to inhibit follicular development and ovulation in the ovary. If she doesn't ovulate, she can't get pregnant. Emergency contraception is for the circumstance where there was unprotected sex, unplanned unprotected sex, or if the condom breaks. So accidentally, sperm got introduced into the vagina and pregnancy is not wanted. She's not pregnant yet, but she doesn't want to get pregnant either. I used to think that that was kind of a lie about the condom breaking. Oh yeah, right, the condom broke. But then it really did actually happen to me once, and I did use Plan B, and I didn't get pregnant. So how does Plan B work? Plan B is available over the counter. You do not require a prescription, and you can walk up to any pharmacy counter and request it. And what Plan B is a pill that contains progestin. And I'll remind you that progestin is a progesterone agonist. It binds to and activates progesterone, and so it mimics progesterone in the body. And so why is this useful in preventing pregnancy? It turns out that progesterone inhibits the secretion of luteinizing hormone from the anterior pituitary. And remember that spike in luteinizing hormone is required for ovulation. So if during this unprotected sex or when the condom broke, if she's in the pre-ovulatory phase of her menstrual cycle, she hasn't ovulated yet, but now there's sperm inside of her, the Plan B, the emergency contraceptive, will prevent her from ovulating, making pregnancy not likely. And if she is already pregnant, because women don't know in the very early stages of pregnancy, they don't know that they're pregnant. If she's already pregnant, this progestin has no harmful effects on the developing embryo, which is why it's available over the counter. I think no one's morality is offended by this as long as it doesn't injure an existing pregnancy. In another scenario, she finds out that she's pregnant and she doesn't want to be pregnant. What are her options? Well, abortion is an option. And there are two basic types of abortion. There's the surgical abortion, where the embryo is physically removed from the uterus, and you go to a physician's office or a clinic to have this procedure performed. And the other category is known as a chemical abortion. And you don't need to go to a facility, but you do need to take pills in order to chemically induce abortions. And these substances, these drugs are called abortifacients. They're drugs used to cause an abortion. And a commonly used abortifacient is called RU486, and it's mifepristone. And how this drug works is by acting as a progesterone antagonist. So I hope you remember that an antagonist will bind to the receptor but block it, and it will not activate it. So this drug blocks progesterone receptors, and the cells respond as if there's no progesterone present. So it mimics a significant decline in progesterone levels. And what happens in a normal, typical female reproductive cycle when progesterone levels fall? Menstruation, menses happens. And so under the influence of mifepristone, she'll get her period. So the implanted embryo, along with the stratum functionalis, the endometrium, gets shed, and she will no longer be pregnant. I make jokes about my adult life's intent to avoid being pregnant, but some women work really hard in order to get pregnant, really, really hard, and other women experience happy accidents and everything in between. Our goal is to let the women who want to have babies, and let the women who don't want to have babies not have babies. That said, amazing things happen during pregnancy. And amazing things happen during implantation. She has no idea that she's pregnant. All of this is happening below her conscious awareness. And a pregnancy begins with implantation. During implantation, the very early embryo, this tiny ball of a few dozen cells, makes its way into the uterus and attaches to the endometrium. And at this point, the embryo, let me pick the color green. At this point, the embryo consists of two different types of cells. The trophoblast cells make up the exterior coating of the embryo, and then the other cells are called inner mass cells here. During implantation, the trophoblast cells that come into contact with the endometrial cells begin to secrete digestive enzymes. They manufacture these protein digesting, these lipid digesting enzymes, and they secrete them, they release them onto the cells that form part of the endometrium. And these digestive cells kill and digest away the cells, some of the cells that make up the endometrial lining. And these trophoblast cells continue to divide by mitosis. And these are called invading trophoblast cells, and they will give rise to the fetal portion of the placenta. So the placenta is this temporary organ. It only serves a purpose during pregnancy, and then following labor and delivery, I guess as part of labor and delivery, the placenta is expelled and it no longer serves a purpose. But during pregnancy, it's an amazing organ that forms from fetal tissue that merges with maternal tissue to form this site for the exchange of nutrients and gases and also certain classes of antibodies. So the placenta is an organ because it satisfies the definition of an organ. It consists of 2 or more types of tissue. And these tissues are interwoven, fetal tissues and maternal tissues. And the placenta develops as blood vessels that arise from fetal tissue penetrate further into the endometrium. And at the same time, those fetal blood vessels are being met with new maternal blood vessels. So the fetus has blood vessels that grow into the endometrium, and the mother's endometrium have blood vessels that grow toward the fetus. And so this interweaving of fetal and maternal tissue form the placenta. So this fetus is obviously much further along in the pregnancy. So this developing fetus has a head and a tail. There are tissues, organs, and organ systems. And remember that all cells in a living thing need to exchange materials with its environment. All cells require gas and nutrients, and all cells need to eliminate wastes. Well, how is that accomplished in a developing fetus? Which organ systems are not functioning in this developing fetus? Which organ systems are not functioning? Is the cardiovascular system functioning? Does it have a heart? Does it have blood? Does it have blood vessels? Is that heart pumping the blood through the blood vessels? Yes. The cardiovascular system is circulating blood. Remember, all of the cells in this developing fetus need oxygen, nutrients, and are generating wastes that need to be carted off. The organ systems that are not functioning in this developing fetus are the organ systems where exchange occurs with the environment. So you remember our sites of exchange with the environment include the respiratory system. That's how we exchange gases with the environment. The digestive system, that's how we obtain nutrients from the environment. And the urinary system, that's how another way in which we excrete wastes into the environment. So in this developing fetus, the respiratory system, the digestive system, and the urinary system are not functional because those systems aren't in direct contact with the environment. So how is gas exchange accomplished? How does this fetus receive new nutrients? How does this fetus eliminate wastes? Well, the maternal blood or the maternal cardiovascular system, digestive system, urinary system, respiratory system needs to take care of those functions for the fetus. And of course, the mechanism of exchange is blood. So here we see a fully formed placenta, and it is firmly anchored to the uterine wall. And the placenta is physically connected to the fetus through the umbilical cord, which attaches to what is now your belly button. So if we blow up a chunk of the placenta, we can learn a little bit about its structure. Well, the placenta is formed from tissue from both the mother's body as well as tissue that arises out of the fetus's cells. And here we can see a merging, an interweaving of the two cardiovascular systems. So in the maternal portion of the placenta, we see arteries and veins, and this artery delivers blood into this maternal blood pool, and this maternal vein drains it away back into her full-on cardiovascular system. And then here in this maternal blood pool, we have this mixing of arterial and venous blood. And this mixed blood pool surrounds fetal blood vessels here. These are chorionic villi. These are fetal blood vessels, and exchange occurs here. So there's no mixing of maternal blood and fetal blood, but rather maternal blood comes to surround fetal blood vessels, fetal capillaries, and that's the site of exchange. So this fetus is able to obtain oxygen from the mother's blood. It's able to obtain nutrients from the mother's blood. It also obtains antibodies, certain antibodies from the mother's blood. And this fetus is able to give off wastes to the mother's blood, wastes like carbon dioxide, wastes like bilirubin to the mother's blood. And then those wastes get processed by the mom's body in the usual locations, right? Her respiratory system is going to exhale that carbon dioxide. Her digestive system and her kidneys are going to help get rid of that bilirubin, for example. So the placenta has two primary functions. One we were just talking about. The placenta is the site of exchange between the mother's cardiovascular system and the fetus's cardiovascular system. Notice that maternal blood and fetal blood don't ever actually mix, but there is an exchange between these two fluids. Oxygen, nutrients, carbon dioxide, other wastes, and antibodies are able to pass between mother and fetus at the placenta. And then a second role of the placenta is to produce hormones. The placenta is quite a sophisticated endocrine structure, and it produces many different hormones that are involved in pregnancy as well as the onset of labor. So parturition is the act of giving birth, and these are the basic stages of childbirth. And notice that the fetus is head down. Its head is against the cervix, the inferior region of the placenta. And at the onset of parturition, that cervix begins to dilate. The aperture gets wider, the opening gets wider, and the amniotic sac ruptures. So the amniotic sac is a fluid-filled sac that surrounds the embryo, the developing fetus. And that ruptures and water passes through the vagina. Water, amniotic fluid, it's not water. It's a body fluid, so it's a dilute watery solution. And that amniotic fluid passes out the mother's vagina, and she has conscious awareness that her water broke. And as parturition proceeds, labor begins. And during labor, the smooth muscle cells in the wall of the uterus contract. And that narrows, it restricts the diameter of the lumen of the uterus. And that applies pressure to the fetus and jams the fetus into the cervix. And now I feel like this is full circle back to when we first started talking about feedback mechanisms. Early in the semester, we used labor and delivery as an example of a positive feedback system. And remember that in any feedback system, there are receptors that monitor the controlled condition. There's a control center that receives that information from the receptors. And then there is an effector. An effector is able to bring about a change in the controlled condition. So here, at the onset of labor, the receptors are stretch receptors located in the cervix. And when the uterine wall contracts even just a little bit, it forces the baby's head into the cervix. And those stretch receptors send nerve impulses. They generate action potentials that are sent along sensory neurons to a part of the brain called the hypothalamus. And in response to increasing frequency of action potentials, the hypothalamus causes the posterior pituitary to release a hormone called oxytocin. And the posterior pituitary secretes oxytocin, and that's a circulating hormone, meaning it travels in blood. And among other places, one of the tissues in the body that has receptors for oxytocin is smooth muscle in the uterine wall. And oxytocin binds to its receptor on those smooth muscle cells. And in response to oxytocin, those smooth muscle cells contract. And that's called a contraction. And that generates more force from the baby's head against the cervix. That's going to lead to an increase in the secretion of oxytocin. And so this is a positive feedback loop where the effector drives the controlled condition further away from the set point. So if the controlled condition here is the degree of stretch of the cervix, the effector here is the smooth muscle in the wall of the uterus. And when that contracts, it increases the amount of stretch detected by the stretch receptors in the cervix. All right, so that's labor, and labor increases in intensity. Those contractions increase in frequency and duration and intensity, and their purpose is to expel the fetus through the cervix and the vagina. And then following delivery of the baby, the placenta detaches from the wall of the uterus, and that is also expelled. Here we see that positive feedback system at the onset of labor and delivery. So our controlled condition here is the degree of stretch in the wall of the cervix. And our effector here is the uterine wall, So let's begin down here when those stretch receptors detect an increase in stretch as the baby's head pushes on the wall of the cervix. That causes these stretch receptors to increase the action potential frequency. And so that signal travels up to the hypothalamus, and the hypothalamus receives an increase in action potentials arising from those sensory neurons in the cervix. And in response to that, cells in the hypothalamus cause oxytocin to be secreted from the posterior pituitary. These are neurosecretory cells. The cell bodies of these neurosecretory cells occur in the hypothalamus, and their axons extend into the posterior pituitary. And from these axons, oxytocin is secreted, and it's secreted into the blood. In this circumstance, oxytocin is a circulating hormone. One of the tissues that has receptors for oxytocin are the smooth muscle cells in the uterus. And in response to oxytocin, they increase their contraction. This forces the baby's head further into the wall of the cervix, increasing the stretch. And so here, this sign indicates that it's a positive feedback system. Placentas are fully formed organs, only they have a temporary need. So following delivery of the baby, the placenta is also expelled from the uterus through the vagina. And the placenta is an organ, and it sits in this pan the same way a heart would sit in the pan, or a liver, or a stomach. It's a fully formed organ. And I have a story to tell here. After my niece was born, I took a photograph of her placenta. This isn't that photograph, this is someone else's placenta. And that year I participated in an event called SantaCon. And this is a long time ago. And SantaCon consists of many people all dressed up as Santa going on a pub crawl. So this started many years ago with the Cacophony Society in San Francisco, back when the Santas were jolly, benign people going on a pub crawl. And since then, I think it has sort of devolved into a group of drunk and menacing Santas that are no good for society. So anyway, this particular year, in the year that my niece was born, I took that photograph of the placenta and I attached it to my Santa Claus suit, and I became Placenta Claus. And for some people, when they approach me and say, hey, which Santa are you? And they'd look at the image, some of them would get grossed out and others would be like, wow, that's really cool. And then I would tell them about the placenta. Humans are mammals. And one of the criteria for being a mammal means milk production. Human parents feed their offspring by making milk for them. That doesn't happen in any other taxon. So lactation, like lactose is milk sugar, lactation is the secretion of milk by mammary glands. And mammary glands occur in the breast. And when she's not pregnant or lactating, those mammary glands are inactive and they're relatively small. But during pregnancy, a whole suite of hormones are responsible for activating that mammary gland tissue. So prolactin is a hormone that's secreted by the anterior pituitary. So mom's anterior pituitary makes prolactin, a signaling molecule, which is responsible for helping to activate those mammary glands. And then there are many other hormones that are produced both by mom's body as well as the placenta that contribute to this activation of the mammary glands. When I say activation, I mean that the cells undergo mitosis. So she'll develop new mammary gland cells, and those cells will increase in size, and those cells become prepared to, and they eventually generate each of the components of breast milk. And it's a complicated thing, breast milk. And if you look at breast milk from any mammal, it's a very complex mixture of all the biological molecules that are required to sustain the life and promote the development of the offspring. This is an artistic representation of a breast, and we're focusing on changes that happen in glandular tissue in moving from the state of pre-pregnancy through pregnancy and post-weaning. So up at the top of the image here, we can see the changes that occur in the mammary tissue in transitioning from not pregnant to pregnant. So the mammary cells undergo mitosis, they divide, and they further differentiate, they get larger. And during the pregnancy, they begin to secrete the components that make up breast milk. And as you can see, these glands are connected to the nipple through a series of ducts. And so during pregnancy and even after pregnancy, while she's breastfeeding, these mammary glands are fully developed and actively producing the wide array of components that we find in breast milk. And then to wean a child means to transition that child from feeding from the breast to not feeding from the breast, to taking a bottle, for example. So when that mother has finished breastfeeding, the mammary tissue once again reverts to the more dormant, inactive state until she gets pregnant again, or if she gets pregnant again. In panel A, in the panel on the left, we can see the basic structure of a breast. So a breast includes adipose tissue, fat tissue. It includes connective tissues known as suspensory ligaments. It includes glandular tissue. Those are the cells that produce the components of milk. And then it includes ducts. Those ducts are little tiny tubes that allow the milk to travel toward the nipple. And the nipple has holes in it to allow for the passage of milk. If we look at panel B on the right, we can see the very basic structure of mammary glands and milk ducts. So these cells here, these epithelial milk secreting cells, each of these cells makes some selection of the rich variety of molecules that are present in breast milk. And then we also have modified smooth muscle cells here. They're called myoepithelial cells, and they are contractile. That's what the myo means. And so they're like modified smooth muscle cells. And when they contract, they narrow the diameter of this lumen, and they forcefully expel the components of breast milk through a series of ducts. And eventually these ducts all lead to the nipple. The milk ejection reflex is also known as milk letdown, and it is a reflex. And we've talked about reflexes in general. We've talked about specific reflexes over the course of the semester, and any reflex has those same 5 components, right? There's a sensory receptor, there are sensory neurons that deliver information to the site of integration. The integration center generates motor outputs, and there is communication between the integration center and the effector. And that's also true for the milk ejection reflex. So there are multiple sources of sensory input here, including tactile sensations, so the mechanoreceptors, the touch-sensitive cells in the breast play a role. Higher brain centers, maybe she hears a crying baby, or she smells something that's like her baby. She thinks about her baby. Those are all sensory inputs for this milk ejection reflex. And the site of integration is the hypothalamus. And the hypothalamus can respond by sending signaling molecules out through the posterior pituitary. So oxytocin is technically, it's a neurohormone. It's secreted by neurosecretory cells whose cell bodies reside in the hypothalamus and whose axons reside in the posterior pituitary. So this is another role for oxytocin. Oxytocin plays a role in signaling the effector in the milk ejection reflex. The effector here are those myoepithelial cells that surround those glandular cells in the mammary gland. Under the influence of oxytocin, those myoepithelial cells contract and help expel breast milk into that system of ducts and out through the nipple. So a nursing mom could hear a baby cry and milk will be ejected from her nipples. And this is called milk letdown commonly, but it's also known as the milk ejection reflex. We can use this image to better visualize, to better understand the sensory inputs as well as the motor outputs in the milk ejection reflex. So in the panel on the left, we see a woman actively nursing her baby. And so some of the sensory inputs here arise with the tactile sensations of the baby on her breast, and that sensory input, along with others, will converge on the hypothalamus, and some of those other sensory inputs are the smell of her baby. She's looking at her baby, she's thinking about how beautiful her baby is and how much she loves her baby, and all of those sensory inputs cause the hypothalamus to generate motor output. So the way the hypothalamus controls the posterior pituitary is quite directly. So take a look at this one neuron. The cell body of this neuron, it's technically a neurosecretory cell. The cell body of this neurosecretory cell resides in the hypothalamus, but its axon extends through the infundibulum and terminates in the posterior pituitary. So with the arrival of all of these sensory inputs, this causes the hypothalamus to increase the frequency of action potentials sent to the posterior pituitary. And cells in the posterior pituitary secrete oxytocin. And this oxytocin is going to cause those modified muscle cells in the mammary glands to contract to help with milk ejection. Another hormone also gets secreted, but this hormone gets secreted by the anterior pituitary. And the hypothalamus controls the anterior pituitary in a very different way. This is a neuroendocrine cell in the hypothalamus. Notice that this is a blood vessel here. The hypothalamus controls the anterior pituitary by secreting, releasing, and inhibiting hormones. So in response to this variety of sensory inputs, this neurosecretory cell in the hypothalamus is going to secrete prolactin releasing hormone, which then travels via the blood to the anterior pituitary. And in response to that releasing hormone, cells in the anterior pituitary are going to secrete prolactin. And prolactin is not necessarily so important for milk letdown, but prolactin is responsible for maintaining milk production. So it maintains those mammary glands in their fully developed and active state. And that brings us to the end of the reproductive system, specifically segment 2. We've just finished our coverage of the female reproductive system. And I have a book to recommend to you. It came out this year and it's called The Vagina Bible. And I highly recommend it. It is a reference book. It's also written in humorous fashion by an authority. Jen Gunter is a gynecologist and she's also a writer. She has a blog and now occasionally she writes in the New York Times, and she's a gynecologist an obstetrician at UCSF. And she writes from a position of authority, from being open to questions. She writes about her experiences as a healthcare provider, and she debunks some common myths and just provides straight up information. And I'll recommend that book. It's called The Vagina Bible by Jen Gunter. And I'll recommend it for anyone who has a vagina or spends any time near them. All right, and with that, I will sign off from lecture for the rest of the semester. Thank you for being here. Take care.